Hello, everyone, and welcome to today's webinar. My name is Olga Bodie, and I'm a SYNGAP parent and part of the team here at SYNGAP Research Fund. Our presentation today is involvement of the brain endothelium in neurodevelopmental disorders like SYNGAP1. I have the pleasure to introduce today's speaker, Dr. Baptiste Lacoste. He's Associate Professor and Scientist at the Neuroscience Program at Ottawa Hospital Research Institute. Dr. Lacoste is a neuroscientist and vascular biologist investigating the cellular and molecular mechanisms underlying metabolic and vascular abnormalities in neurodevelopmental disorders. His lab revealed that the NDD mouse models display early neurovascular abnormalities caused by endothelial dysfunction, leading to brain metabolic imbalance. He also co-led two recent studies showing that perinatal stress, hyperoxia, and high-fat diet triggers long-lasting cerebrovascular and behavioral alterations reminiscent of autism. Dr. Lacoste's research career started at the University of Bordeaux in France, where he developed his passion for neurobiology. He left France to pursue his graduate training at the University of Montreal, 2003 through 2009, and his MSc PhD work allowed him to acquire solid expertise in neuroanatomy and neurobiology, while investigating the trafficking of substance P receptors using pharmacology and electron microscopy. He continued his training with the first postdoc position at the Montreal Neurological Institute, McGill University, where he developed a new passion for vascular and continued with a second postdoc in Boston at Harvard Medical School. Dr. Lacoste examined the influence of neural activity on cerebrovascular patterning during postnatal development, as well as the mechanisms governing blood-brain barrier formation and integrity. Along his training, Dr. Lacoste thus established an original research niche based on the conviction that studying neurovascular interactions in the healthy and diseased brain is a very promising avenue in neurobiology at both basic and translational levels. A recorded version of this webinar will be available on the SRF website under webinars on the family menu. And by the end of this presentation, you will have the opportunity to get the answer to your questions. We'd love to hear from you, so please put those questions in the Q&A below. And for those of you just joining us, welcome. And again, our speaker is Dr. Baptiste Lacoste, and his presentation today is involvement of the brain endothelium in neurodevelopmental disorders like SYNGAP1. It's now my pleasure to turn things over to Dr. Lacoste.
Thank you very much, Olga, and thank you to the organizers for having me today. So without further ado, we're going to dive into what we've been doing in the last three to 5 years. And as the title says, we're going to look at vascular factors in autism spectrum disorder, which is a new angle we're developing, in which we hope to apply to SYNGAP research, of course. So let me give you some introduction concepts first. So why is it so important and interesting to us to study the brain vasculature itself? Well, I'm going to just go with some facts. First of all, the brain, which is somehow a small organ compared to the size of the body, in fact, consumes a lot of the energy which is available at rest. So people tend to say that 20 to 25% of the cardiac output or the energy available is targeted to brain energy consumption. So as we say, the brain is very hungry and it needs a lot of energy constantly for functioning. And unfortunately, as opposed to muscles and fat and other type of tissue, the brain doesn't have a huge capacity to store the energy. So it's an organ that needs a constant supply of energy. And the only way to bring energy, and when I mean energy, I mean nutrients such as glucose and oxygen, for instance. Well, the only way to bring energy to the brain are the blood vessels, because blood flow is actually the vehicle for oxygen and nutrients. So when people study, like me, like us, I should say, the brain vasculature, we usually tend to divide in 3 big domains, which we are going to see some of it today. But first of all, the brain has to develop sufficient amount of blood vessels to be perfused in an efficient manner all over the place, because the brain has so many different regions. And this is a process called angiogenesis, or I mean, in order to ensure that vascular patterning, there is a sufficient numbers of blood vessels and vascular density across the brain for sufficient perfusion. The other large, big field is the field of the blood-brain barrier, which basically says that the blood vessels of the brain are very special compared to other organs, such as the kidney, the lungs, in which they are very tightly sealed by particular junction between cells and selective transporter. So I don't want to go into details, but this is ensuring that the brain is always working in a safe environment for neuronal function. And this is what we call the regulation of brain homeostasis. The third big theme of neurovascular research is how the blood flow is regulated, such as, for instance, the needs in energy are met by the supply. So this is what we call the regulation of cerebral blood flow. We'll come to it in this presentation. There is even a more nerdy term that we call neurovascular coupling, through which increases in neuronal activation drives increases in blood flow in different regions of the brain to ensure that there is a proper functioning of neurons. And just to give you a side note, if ever a region of the brain lacks perfusion, blood perfusion, this is leading to what we call a stroke, in which neurons are going to die because of the lack of oxygen and glucose, and neurons in the core of that injury die very rapidly, and they are surrounded by a zone which we call the peri-infarct zone, which is fragile and needs to be saved in an efficient and rapid manner. So this is just to say that in order to have a maintained brain health, brain development, brain function throughout life, you need healthy blood vessels. And from development, from the phases where these vessels are growing and installing into the brain, to adulthood and aging, during which, well, vascular health needs to be maintained. So that's what I'm kind of giving you this image of a guy working on a tight equilibrium, because many of the conditions I'm listing here are actually affecting directly blood vessels and hence affecting directly brain development and or function. So in front of us, we have two classic examples, which are perinatal stress, hypoxia. So we've been involved in collaboration working on these themes. Of course, stroke, lack of supply of energy is going to kill the neurons right away. But we have also mutations, which can lead to malformations, which can lead to delayed maturation and delayed functioning. And we also have collaborated with different groups in Canada to study maternal immune activation, showing that perinatal stresses, including inflammation around birth, around and before, can lead to long-term neuronal defects and actually lead to behavior reminiscent of autism spectrum disorder. So affecting blood vessels throughout life is actually leading to conditions that some of you might have heard of: vascular cognitive impairment, dementia. Later in life, actually, Alzheimer's disease, which is a neurodegenerative disease, is more and more recognized as a vascular disease. Multiple sclerosis, which affects the blood-brain barrier and induced leakage and entry of immune cells to the brain. Cerebral palsy, which is in fact a pre, or postnatal vascular injury, such as a stroke, leading to paralysis of part of the body. And we are bringing this new dimension, which is in fact that early vascular deficits in life, whether it is during pre or postnatal development, can actually lead to autism spectrum disorder. So, so that's going to be the theme of today's webinar. And I'm still going to go a little bit deeper in introduction concepts, because I think this is very important for the data, which is following up. So as I just said, there needs to be an equilibrium between vascular health in order to ensure neuronal health throughout life. So that's said. Now, what are these blood vessels? So there is another thing I would love you to remember after today's presentation, is that blood vessels, which can look like this image in the center, are not just passive pipes and tubes that bring blood and oxygen through the brain. In fact, if you do a section of the blood vessel, you'll see that they are composed of many cell types and different elements. So the whole unit of vascular cells and perivascular cells is named the neurovascular unit. So I'm going to quickly mention a few of the important cell types that are present around these vessels. So first of all, the wall itself, the tube itself, and we'll see that it's not just a passive pipe, is made of endothelial cells. So these cells, which are epithelial cells, but since they are found inside the vessels, we call them endothelial cells, are lining up the wall and actually are making very specific junction between each other, which are called tight junction, ensuring that the vessels are pretty impermeable. Just past the vascular lining of endothelial cells, we find the pericytes at the levels of small vessels in the brain that have many functions, including regulation of the blood-brain barrier and neurovascular coupling. So I won't touch too much on these cells. And around those cells, you can have like those end feet, those extension of the astrocytes, a glial cell type in the brain that are completely covering the vessel wall. Again, for ensuring many different functions, which I'm not going to touch upon today, but past that wall, the nerves, so the axons, which is basically the extension of the neurons, are touching the vessels and communicating with this neurovascular unit to actually regulate the blood flow. And this regulation of blood flow is ensured partly through contraction and dilation of the vessel, which ensures regulation of the speed of blood flow inside. So if the neurovascular unit is affected in a way or another and in different parts or throughout lifespan, well, neurons are going to be affected. And that's, again, what I'm going to touch upon during this presentation. And the health of the neurovascular unit is extremely important as it ensures many of the key features of brain health, which are maintaining vascular permeability, ensuring that angiogenesis, this process through which all these new vessels are formed, is happening correctly, regulation of vascular tone, which is, vasodilation and vasoconstriction, brain homeostasis, as we mentioned, through the blood-brain barrier, but also regulation of brain metabolism. So metabolism is a complex word, but it basically means how these nutrients or sources of energy are going to penetrate the brain, are going to be used, they are going to be metabolized, so used to produce the actual source of energy in a cell. There is another very important point before I move on to the data, is something I didn't actually mention so far, is the health of vessels in the brain, particularly in the developing brain, is ensuring proper neurogenesis or proper formation of new neurons. And let me explain in this schematic here, which is taken from a very good review published a few years ago, that is showing maybe one message which is important to retain here, is that the endothelial cells, so these cells that we've seen here inside the vessel wall, are not just cells that are passively ensuring the structure of the tube, but they're also producing different factors, different proteins that are diffusing to these differentiating newborn neurons. So here in this example, we can see that endothelial cells can secrete and also nitric oxide, they can secrete VEGF, vascular endothelial growth factor. They can also secrete BDNF, brain-derived neurotrophic factor, and also different molecules which called neurotrophins, for instance, which means that the endothelial cells can instruct the neuronal progenitor to differentiate, to stay quiescent, to migrate, and to turn into the cells that they shouldn't turn into. So what I mean by this is the health of the endothelium from the get-go is very important for normal neuronal development. So here we go. Now the main question that I aim to tackle in this presentation is, well, how and when and what mechanisms underlie these vascular deficiency early in life and their consequences on brain maturation. So one of the ways to look at how vascular deficiency could affect neurodevelopment is to look at neurodevelopmental disorder. And we decided to focus the lab on autism spectrum disorder. So I'll come a little bit in the next slide about kind of a definition of it. But before we went into developing this idea, we looked around in the literature and found some evidence or indirect evidence that, yeah, the vessels in the autistic brain, or I should say in the ASD brain, may have abnormalities. So we found this study published in 2015 showing that using functional magnetic resonance imaging techniques, there were groups observing that some regions of the brain, such as the cerebral cortex, were either hypoperfused, so less blood flow, or hyperperfused, more blood flow. So that was something which was an indication that something was going wrong. Again, in a global ASD population, so when I say global, means not really stratified by the mutation causing the syndrome, they use post-mortem brains, so after death of a population of young autistic patients, in this case, actually from, I think, 8 to 28 years old, if I recall it properly. But actually looking at sections of those brains post-mortem, they actually observed that the vessels looked different. They looked more tortuous. They look actually, they had more pericytes around them at a stage where pericytes should have stopped their proliferation and other features. But these were indirect observation because it's in a fixed brain, post-mortem brain, but still something was different about those brains. And the same year, in 2016, another paper published an interesting discovery that if in a specific neutral amino acid transporter, which is the name coding for this transporter is called the SLC75, was absent from the endothelial cells of the brain, this would trigger autism-like behavior in mice. So in fact, it meant that lack of this amino acid transport through the blood-brain barrier was causing a defect in neuronal maturation and hence showing behavioral abnormalities. So actually, this motor delay, motor coordination problem that they observed in these mice was rescued, repaired by bringing back this missing amino acid. So as you see, there is something about the vasculature. However, the main question that we aimed at tackling when we started this set of studies was, where are these cerebrovascular abnormalities, structural and or functional determinants in ASD pathophysiology, whether it is disease onset and or disease progression. So to test this, we decided to choose a good model of autism spectrum disorder. And I would say even more, a good model of neurodevelopmental disorder, because when you find something in one specific model, it doesn't necessarily mean that it's generalizable. But if you take a model that is a most common mutation, let's say found in autism spectrum disorder or neurodevelopmental disorder, that becomes to be quite interesting. So a quick definition, but of course, that's something that this audience may know, that autism spectrum disorder are defined by deficit in social interaction, speech and language delay, communication problems, and cognitive impairment. But that's something which is maybe too simplistic summary. But this actually is found in about 2% of kids worldwide. And an important fact is, well, we always talk about genetic mutation, but of course, our neurodevelopmental disorder are a mix and an interaction between environmental factors and genetic susceptibilities. But we decided to focus on the 16p11.2 deletion syndrome, which is the most common mutation causing neurodevelopmental disorder in humans. And in terms of pure autism, diagnosed autism, it is found in 2 to 3% of these cases. But it causes many features belonging to the ASD, such as developmental delay, intellectual disability. They also have metabolic disorder, increased prevalence of seizures, head size is different, and so on and so forth. But the most important thing when you study this in a lab setting with animal models, for instance, in our case, is to find a good model that is faithful to the human disease. And that exists actually. So people in the lab of Alia Mills in Cold Spring Harbor has developed a mouse model of the 16p11.2 deletion syndrome. And this deletion, I'll show you a little diagram how it looks like, it causes haploinsufficiency of about 30 genes, meaning that, in fact, if you looked at, so at the bottom left image here, on the top, you will have a drawing, a schematic representation of this region of the human chromosome on the top here, human chromosome. And on the mouse chromosome, actually those exact same genes that are found in the locus 16p11.2 are found in mouse locus 7qf3. So meaning that the same exact genes in the same order are found on chromosome 7 in mice. So this is what we call a region of synteny conservation. So what this lab has done actually is removing half of these genes, meaning on one copy, you know that in the genome, we have one copy from mom and one copy from dad, and then it actually caused an heterozygous mutation, leading to, and that's what we see on the bottom right here, an haploinsufficiency for this gene, meaning that at this locus, the expression of these 27 to 30 genes is reduced by 50% in the whole body. Okay, so these mice actually are very interesting because they show hyperactivity like human patients. They show repetitive movement. They have difficulty to adjust to new environments, which is another specifics of this disease. Social interaction deficit, like in ASD, motor coordination problem. They also have very strong male-biased deficits, including a sleep cycle change. However, in our studies, what we aimed at focus on is the vascular features. And so if you're interested in reading more details about this study, we published it in 2020, so in Nature Neuroscience. So I'm going to summarize it basically for you now. And we started by asking the question, do these mice, or these 16p11.2 deficient mice, display some of the features that have been found in human populations of autism patients, such as regulation of blood flow, and these differences we saw in hypo or hyperperfusion of the brain. So in this image here, what we did is injecting a tracer, which is called a microbubbles, tiny particles that are detected by ultrasound. And after injection in the periphery, they go to the brain, and we can detect them using ultrasound. And what this graph is showing, to make a long story short, is after a certain amount of time where they penetrate the brain, we see that at the baseline value in a mouse that has been anesthetized, basically, the perfusion of the brain is higher. And this was measured in the cerebral cortex, the superficial part of the brain. So, well, as in some human studies, this mouse shows a higher perfusion of the brain at baseline, at rest. But so it doesn't mean that this is due to vascular feature. It could be to maybe there is more neuronal activation, and you remember this concept of neurovascular coupling. When there is more activation, there is more blood flow. Well, to kind of dissect the contribution of neurons and vascular cells to this apparent phenotype, we looked at neurovascular coupling, and we stimulated neuronal activation in the cortex of these mice by, in fact, stimulating the whiskers of the mice and measuring blood flow in the corresponding region of the brain, which is called the somatosensory cortex. And what we observed is that during whisker stimulation, which is the region in yellow here, the increase in blood flow due to increased neuronal activity was of lesser amplitude in the 16p deficient mice. So what does it mean here? We have in front of our eyes here a defect in neurovascular coupling. This also could be due to changes in neural activation. So I'm not going to go into the details of this because it's in the publication, and I might not have time for giving you all the details. But we actually found that this lower response, blood flow response to neural activation in 16p deficient mice was not due to changes in neural activation. So what we did is we isolated vessels from the brain, or vessels from the superficie of the brain, which is called the middle cerebral artery. And this is an artery that we can actually isolate, put under a microscope, and apply compounds on it, such as molecules that are known to induce vasodilation, or other compounds that are known to induce vasoconstriction. And what I want to summarize in this top right corner here is, in fact, when we applied acetylcholine, which is a vasodilator, which in fact needs a healthy endothelium. So I will pass on the detail on how this is working, but what we can see in this image is the more you increase acetylcholine concentration around that artery, which has been isolated, you should, in the normal wild type artery, have a strong vasodilation. You see, this is a % induced vasodilation here. But in the 16p deficient arteries, vasodilation to acetylcholine, which is an endothelium-dependent vasodilation, was not really happening. And the other panel here shows basically the same thing. This is a defect in the endothelial response to acetylcholine. So this was telling us, well, all those features here are definitely reminiscent of vascular deficits. And well, since this vasodilation to acetylcholine is endothelial dependent, and since we are working with a neurodevelopmental disorder mouse model, well, we looked at the development of vasculature in the brain of these mice. And what we saw was actually quite surprising is at younger ages, so all these physiological measures that we have here were taken in adults, P50s, postnatal day 50. And we went earlier in teenager mice, and we saw that they actually had less vessels in the cerebral cortex. You see that in this image, we see less reduced density, which is calculated here. So then we said, well, there is something wrong with the endothelial cells in the brain of these mice. So to assess function of these cells, we isolated these cells using a procedure that we published in the protocol if you're interested. And then we looked at different things such as their function. And one of the tests we can do is test whether they could trigger new vessels in the dish. So you seed those cells separated in a matrigel, which is a 3D matrix, artificial matrix, and spontaneously, these cells are going to form vessels in vitro. Well, this was not the case with 16p deficient cells, endothelial cells from the brain of these mice. They were not able to form those tubes, and this is quantified at the bottom here, meaning that they were not able to do what we call sprouting angiogenesis, forming new vessels from existing nodes of vasculature. So this was surprising, but this was independent of their proliferation rate. This is measured through cell cycle analysis, and these cells were proliferating normally. We actually have in the article, they actually migrate normally, but they were not able to induce angiogenesis. So we found this very interesting, but this is not sufficient to say, well, this might actually be also found in human patients. So what we did was replicating the exact same experiment using endothelial cells that we induced from pluripotent stem cells from patients that have the deletion. So what we do is we take what we call iPS cells, we turn them into endothelial cells through a complicated protocol, and then we can, first of all, like you see on the bottom left here, confirm they became endothelial cells, and then replicate this in vitro angiogenesis assay. And exactly as in mice, this human endothelial cells that have the mutation, the same exact mutation, didn't form those tubes, showing that, yeah, this is conserved in humans. This is a phenotype that is also found with human cells. And I would say that most interestingly, this first study finished by a conclusion that not only these 16p deficient mice show defects in brain endothelial cells, but this endothelial defect triggered by the 16p deletion were sufficient to induce behavior in mice that are reminiscent with autism spectrum disorder. And why do I say this? Because in fact, what we did is engineering mice such as the 16p deletion was only present exclusively in endothelial cells, leaving intact all the neurons and all the cells of the brain. And we found that these mice with an endothelial specific deletion also showed hyperactivity during the night phase, which is measured by a Bing break assay. And here, they also showed, like the full mutant mouse, the constitutive mutant mouse, a different behavior in what we call the Rotorod test. So here you see that they learn the task and they actually perform, I would say, perform better than the normal mice, which in fact, it means they have difficulties to learn the task and also show hyperactivity. So the conclusion was, well, endothelial defects participate to the onset and progression of this particular syndrome. But the next question was, and I'm checking time here, yeah, okay. The next question was, okay, well, we know that 16p deletion or 16p haploinsufficiency triggers endothelial, brain endothelial deficits. But then the question is, what is wrong with these cells, such as they don't do what we ask them to do? Well, we tackle this question by looking at the energy function of these cells or the metabolic function of these cells. But because we think that, well, at the time we thought, now we have the answer, but we thought that if these endothelial cells cannot do the stuff we ask them to do, for instance, form tube in vitro, induce vasodilation, and so on and so forth, is maybe they lack the energy to actually do these functions. So we measured the metabolite concentration in these primary mouse brain endothelial cells from wild type versus 16p deficient mice. And what we found actually is all the metabolites, almost all, and particularly those associated with energy metabolism, were all reduced. So this is a heat map showing basically that, so we did in 2 cohorts because we really wanted to repeat and see whether it's true, depending on the batch. But if we concentrate on the right panel here, you will see that the colors are darker for the wild type mice, meaning that all the metabolites that are found here are at lower concentration in 16p deficient endothelial cells. So for now, the conclusion is, well, there is a lower metabolite concentration. But what are those metabolites which are particularly decreased in these mice? Well, we were very surprised to see that metabolites involved in energy regulation, energy state of the cell, were all reduced. And the most reduced included ATP, adenosine triphosphate, which is the main energy source for the cells and all the cells of the body. So this ATP was reduced by more than 50%. So here you have a cell that is in the mouse, that is alive, that ages well, that is, I mean, healthy, if you look at it globally. But in these brain endothelial cells, ATP, the main source of energy, is reduced by more than 50%. So we measured a bunch of things, including the oxidized versus reduced glutathione ratio, which is indicating that in this case, since this ratio is not changed, that the cells at the time we extracted them were healthy. But each individual element of this ratio was also reduced. Also reduced the adenylate energy charge and the ATP to ADP ratio. So everything is basically saying that in these brain endothelial cells from 16p deficient mice, there is an energetic failure. So I'm going to pass on this, but it would have been expected that activation of AMP kinase goes up with a low and very low ATP concentration, but it barely reached the significance. So the main thing, well, when we see this energetic failure, is to look at mitochondria. Mitochondria being the battery of the cell. And we did a classic seahorse test. And usually when you see this the bioenergetic failure, you should see a reduction in mitochondrial function, which we didn't see. So there is something else going on here. All the parameters here, including ATP production by individual mitochondria, was not affected. So this test, to bear in mind, is not sensitive to small changes in mitochondrial number. So this is what we next quantify. Maybe they have less mitochondria or they have less numbers, and that's actually was the case. When we did, first of all, in vitro, culturing these endothelial cells, staining for mitochondria and quantifying the intensity or the density of this mitochondrial network, we actually found a reduction, significant reduction in 16p endothelial cells. And this was observed at P14, so 14 days after birth. But if we take adult mice, and now we go in vivo using electron microscopy to count mitochondria, well, we indeed found a reduction in mitochondrial number. So in fact, these endothelial cells from 16p mice have less batteries in the cells. So that's very surprising. And the cells surrounding the vessels, well, did not show a change, which seems to be, again, a vascular specific change. So when people observe reduction in mitochondrial number, it could be two things. We have to check as whether, well, are these mitochondria more fused together? So you count less doesn't mean that they produced less. Maybe they have fused more into larger mitochondria. But in fact, we actually, so I don't show it here, but the morphology of these mitochondria was not changed, first of all. But also, if you look at all these proteins, so OPA1, DRP1, MFN1 and 2 are proteins that are regulating mitochondrial dynamics, which is basically meaning that it regulates the ability of mitochondria to either fusion or separate, which we call mitochondrial fission. Well, all these proteins were not changed. Whether we looked into whole cortex extract or primary brain endothelial cells extract. So no change here. However, we were very surprised to see that a transcription factor, which is essential for the formation of mitochondria, was not reduced, but lacking in these endothelial cells from 16P mice. So basically, we usually look at PGC1 alpha. It's an transcription factor regulating mitochondrial biogenesis, so the production of the formation of mitochondria. The full length PGC1 alpha was barely changed. However, for some reason that we don't know yet, the N-terminal splicing variant anti-PGC1 alpha was completely lacking. And I'm going to show you the original Western blot in this case. Well, in the mutant mice or in mutant endothelial cells that were extracted from the brain of these mice, we don't find this isoform, which is very surprising. So that's one thing that we're going to pursue and, of course, use as a rescue for the base of rescue approaches. But there is something I want to show you because we did a weird experiment, the type of experiment that the boss asked you to do, but the reason behind it are not clear. But basically, it was a naive idea to say, well, we have less ATP in these cells. Why not adding ATP to the dish in endothelial culture and do the matrigel assay, which is testing endothelial function? Well, Julie and Nicole's students in the lab actually added ATP in different concentration to endothelial cell culture and did the matrigel assay, and it completely rescued the formation of tubes, saying that definitely if you bring back more ATP, you can actually correct these deficits completely. So that's a proof of concept study that we need, of course, to dive more in because ATP was added outside the cell and we don't know if it goes in or if it acts through a receptor. We also have experiments in progress where we actually bring ATP inside the cell through different processes. But of course, the main idea now is going to be to try to rescue expression of this transcription factor to bring everything to normal, hopefully. So one of the things I'd like to finish with is, which belongs to a study we just published, is that not only these brain show endothelial deficit, and not only these endothelial deficit can trigger behavior that are reminiscent of autism spectrum disorder, they also trigger a complete shift in the way the brain uses its energy. So that's what I'm going to show you quickly in this image here is we did an experiment which is assessing glucose uptake, which for some is an index of brain metabolism, but some other is more just for how much glucose is pumped into the brain. This is actually used in humans regularly. So what we did is injecting an analog of glucose, which is called FDG, fluorodeoxyglucose, which is radioactive, and you wait, and then you see how much has been pumped into the brain. And we see, I don't know if you see how striking this is, the 16P deficient mice show a very high increase in glucose uptake. So the whole publication that is just out now is kind of going into what is happening in terms of metabolism. But the thing I really want to finish with today is showing that if you trigger or if you limit this mutation again to only the endothelial cells, you also find this brain-wide metabolic increase. So that is to say that these endothelial deficits are sufficient not only to drive behavioral phenotypes, but also physiological phenotypes such as hypermetabolic, brain hypermetabolism, particularly of glucose in this case. And again, this has just been published, so I invite you to consult this paper. So to finish with, I'm going to give you some take-home message I hope you can keep with you. Is first of all, the 16P11.2 haploinsufficiency, so this deletion, is impairing mitochondrial biogenesis in brain endothelial cells, which is causing this failure in energy production. So this was actually another important finding is that endothelial haploinsufficiency, this deletion, is sufficient to kind of have the brain adapt its metabolism, because in many ways, the way that nutrients, oxygen, and all and different things are going to go into the brain are differing in these cells. And the main message of what we do in the lab currently is, of course, that vascular endothelial cells are key player in pathology of at least this ESD syndrome. But again, nowadays, the way that people study autism is very neurocentric, and I think we should definitely take into account the vasculature of the brain, which is so important. And what is next for us? Well, and particularly concerning SYNGAP1 syndrome, well, we want to apply this knowledge. And first of all, we want to try rescue approaches for 16P and also try these measurements of mitochondrial function, metabolism, and vascular features in models of SYNGAP1 syndrome, such as rodent and cellular models that we are currently acquiring, also thanks to a seed funding we received from SRF. So thank you for this again. And just one thing, that if you go online and you look at RNA-seq databases, basically showing you gene expression of SYNGAP1 in the mouse brain, you'll see that it's, of course, expressed by neurons and astrocytes, but look how much it is expressed by endothelial cells in the brain. So, of course, if you remove half expression of SYNGAP1 or the totality of SYNGAP1, I'm predicting that we also will see some interesting functions and phenotype. So, of course, this work is not possible without a fantastic team of students and trainees. The leaders of this project are Julie, PhD student in the lab, Nicole, PhD student also, Alexandria, who just published the survey part and has moved over to new adventures and was a PhD student, and all the others. And of course, I thank all the collaborators and the funding and you for your attention. And I'm ready to ask any question you have. Thank you.
Thank you very much, Dr. Lacoste. That was absolutely fascinating. And I think the audience is a combination of parents and scientists. So if anyone wants to put questions in the Q&A, or if they want to raise their hand, if you want to speak, it's a nice group we have here. So let's just let it rip. I'm glad we were able to get you some cell lines, and I will tell you, there are a couple of cool patient-derived mice that should be published pretty soon too. So in addition to the resources you're getting in Canada, hopefully there'll be even more at the JACS. But
wonderful.
I'm just struck. I'm not a scientist. I wish I was, but I'm just struck hearing your talk about. I think many of us, and this has been confirmed in some interviews for some studies, when our children, because Syngapians are characterized by, at first, large amounts of atypical absence seizures, which includes eyelid myoclonia, and then eventually, unfortunately, those progress to drops and drop seizures and other seizure types. But a number of us have said, when our child is actually focusing and trying to do something, puzzle, game, drawing, whatever, when they're actually trying to use their brain, we see them get tired so fast. I'm not talking about hypotonia fatigue. I'm talking about mental fatigue. And then we see the seizure activity kick up. It's a number. I've heard multiple parents describe that same phenomenon independently and unprompted. And hearing your presentation, I wonder if there's some connection there. So I guess the question would be, am I making that up, or am I on the right track?
So in terms of an experienced parent of SYNGAP1, I will not comment because I'm not myself, so I didn't observe firsthand. But of course, what you're saying makes a lot of sense. So, and I'm not a neurologist, so I'm excluding all the non-knowledge first before answering you. But of course, so things that are very interesting are, first of all, that there are a lot of vascular features involved in epilepsy in general and seizures. So a lot of people work on this aspect of epilepsy. And again, nutrient transport, nutrient delivery, regulation of energy metabolism, all these things have been implicated in epilepsy and seizure. So this has never been studied like in any way in the context of autism spectrum disorder, a particularly vascular feature. So that I cannot give you scientific info on this because it has never been assessed. So the interesting thing about brain metabolism is, as you see, so we kind of show you here, and we hope to continue in different syndromes, particularly SYNGAP1, that having a vascular deficit from the get-go is definitely going to influence the brain, of course, develops functions, but also uses energy sources. So what you're telling me is just another reason for me to go and chase after metabolic features. And the key of metabolism is the way that these nutrients are brought to the brain, which is the vasculature. So that's one thing. And I think because you're kind of linking things I wanted to say, I'm adding the fact that SYNGAP1 regulates the activation of a protein we call ERK1. And ERK protein involved in many metabolic regulation is found on the 16p deletion locus. So what I mean is they also might have functional interaction that makes the study of both diseases a very interesting framework for comparing outcomes and phenotypes and making those as models to kind of, tackle completely new question, new biological question. But what you say is very interesting, and it pushes me to think that yes, there might be something about the brain vasculature.
The other thought I had is that strikes me about our kids. And then JR, actually, who's a parent and a scientist, asked a very good question in the Q&A. But the other point I'll observe about Syngapians is their heart rate just goes crazy. Like when they get worked up, their heart is just pounding out of their chest. And I'm reflecting on your first slide where the brain takes 25% of the energy, right? And I always thought it was just an anxiety thing. Not that I understand the mechanisms of that, but now I wonder if it's just the brain saying, get me more food, and the body trying to move the blood as fast as possible. Maybe you can put a heart rate monitor on those poor little mice.
That's very interesting. So again, I'm not going to say yes, this is this, but yes, this is something that needs to be tested. Because so one of the things that you will see in the next couple of months and years is the whole field which is developing in neuroscience is the interaction between heart and brain. So even in Ottawa, we just got a $100 million grant, not me, unfortunately, but the institution, to study brain-heart connection. So part of this money is going to go to different labs, including mine, and we are going to acquire very advanced tools to study those questions. So of course, these models of neurodevelopmental disorder become also part of this stream of studies connecting the brain to the heart. If you have a defective brain, it leads to defective heart, and vice versa. For instance, people who have depression are more likely to get cardiovascular disease. People with cardiovascular disease are more likely to develop mental health disorder. So what I mean is this is something that is in progress, and my lab is going to this direction. And yesterday I was looking at some new technology, very cool in mice, where you can measure both brain activity and heart activity. And that's something we could apply to one model as well.
That's cool. I want to, before I read Janie's question, I want to just observe there, we have a researcher at UPenn named Benjamin Prosser. I don't know if you know about Ben, but his daughter had STXBP1, and he started working on STXBP1 and SYNGAP1, and he's actually become a pretty important researcher in our space. But what's cool about Ben is before his daughter got sick and he started making ASOs, he actually had a cardiology lab. So he could be a great collaborator for heart-brain things. And he knows how to spell SYNGAP1. But so JR's question, I'll let you read it, but I'll read it for the audience. Thank you for this great data and insight. I wonder how the mitochondrial count will look in SYNGAP1. I wonder how many different ways the energy flow can be messed up and how it will be in SYNGAP. It seems very likely. Also adding on to Mike's comment about energy, my son eats a lot and is very underweight. I wonder how much he is using just laying around.
Yeah,
and then
well, that's why, I mean, I'm discovering the field of SYNGAP1, and I'm super excited by these ideas. And you know that I love to be fed with this type of idea, because we're going to test these things. So we have the tools, we're going to get the models pretty soon. But yeah, thank you for your insight. And that might be the case. I don't want to say that everything is due to brain endothelial cells, because I'm going to look like a reductionist. But just looking at the levels of expression of SYNGAP1 in the brain endothelial cells, I can tell you we are going to find differences related to vascular function.
And then before we go to Devika's question, you move pretty quickly through the ATP rescue.
Yeah.
But without being prescriptive or telling us, what are the implications of your work for rescue with small molecule? I mean, you said we threw ATP in a dish. Like, what can we put in a mouse that would get to a brain that could actually help with energy production? Yeah,
so that's an excellent question, and Devika and I actually are developing collaboration on this. So I'm glad she came today. So the reason why I'm kind of I was insisting on the fact that my idea of adding ATP to the dish was a naive idea is because ATP has a half-life of 15 minutes in the dish. It's going to be degraded. It doesn't have to stay at too high concentration around the cell because it can be toxic, but it has also to enter the cell. As I showed, ATP is reduced by 50% inside the cells. So it means that there is a mechanism that we have to now understand and kind of dissect on how this extracellular ATP could very surprisingly rescue. Actually, I've talked to a stem cell researcher here in Ottawa Bill Stanford, which is a well-known PI in the field, we have observed the exact same level of rescue using human-derived cells from 16P patients. And he told me he has never seen a rescue that efficient. So we are onto something. The thing is, ATP treatment in autism, if this is your question, was already kind of proposed. And we had received recently, 1 year ago, funding from the Department of Defense, the US Department of Defense, to test this idea. So we actually are treating mice with ATP and see what it does. And I have a student, I should not talk about the data too much, but I have one student tested on one behavioral test in 16P mice. It's actually rescued by IP ATP treatment, which is crazy when you think about it. But Devika and I now, and because she's developing so much of interesting tools for nanoparticle delivery and extracellular vesicle delivery to bring ATP and other factors such as PGC1 alpha into the cell. So we have grant applications submitted with this exactly. And I'd love to test those rescue approaches with SYNGAP1 as well. Once, as Jenny said, we have assessed the numbers of mito and their function in SYNGAP1 deficient cells in mice.
Got it. Okay, so do you want to take, so I've said Devika, but I should be saying Professor or Dr. Manikam. Her question, have you considered treating the mice with PGC1 alpha activating compounds such as, I'm not going to even try it, a natural anti-inflammatory compound to increase mitochondrial biogenesis?
That's a very cool idea. Actually, restroveridol and what the other name of the of course, and when I look for them. So these molecules are very interesting and used in many vascular deficit or vascular impairment type studies. And we have actually recently used restroveridol and combined with exercise to in a stroke recovery study. So I didn't know that resveratrol can boost PGC1 alpha, so that's very interesting. But Devika and I are actually thinking about, bringing up PGC1 alpha in different ways. So what I mean by rescuing these lack of expression or these deficits, this is exactly the next step the lab and our collaboration is going to go after. That's exact next step, but that's a great idea. You helped me find this MPK activator. Completely forgot the name. Metformin, yeah, sorry. So metformin treatment also was interesting, and we have not tested it. No.
I mean, one thing I will say to you and Dr. Manikam is there are more and more small molecules with hypotheses that are popping up. 6Bio is another one. There's a paper by James Clement, not related to this, but we currently lack a really efficient way, a really efficient assay to throw molecules at. So that's just something to think about.
Actually, I mean, it's a good
lot
point.
of different things.
Devika's lab has published and confirmed expertise in engineering those lipid nanoparticles for delivery systems. So she's the perfect collaborator for me, actually, for trying to treat these mice.
Great. And if my Google skills are up to par, she is in Pittsburgh.
Yeah.
That's great. We have a few patients in Pittsburgh. Really good people. And then JR had a question about the ERK connection in 16P. Yeah. Will you make double mouse cross SYNGAP pathogenic mutants to see if the double is more severe or compensated in some ways, dosage more similar?
Yeah, so that would be a great idea. But first, I would start by something less risky. I would definitely test functional interaction between. I mean, I would confirm the ERK regulation in brain endothelial cells in vitro from mice and from the cells we're generously going to get with support from the SRF. So this interaction is very interesting. Yes, so we're gonna definitely study it. Now, if I understand the question, I think that's a step a little bit further down the road. Since the ERK regulation by SYNGAP1 is already published, I would definitely see if we observe vascular deficits in SYNGAP1 cells and mice. These can be modulated or rescued by modulating ERK activity. So that would be the type of approach I would take as a target. But my lab has a tendency to go unbiased first. And we get this, now we have the proof that vascular features are important. We are data-driven. So we do omics, we do metabolomics, RNA-seq, and proteomics. And from these, we can build networks of functional networks of proteins or genes that are involved and run after some targets, which definitely is one of the things we'll do with SYNGAP1 mice.
Super. Super. Other questions from the audience? This is a very dense and wonderful webinar. I think a lot of us are going to chew on this. I have a hand up from the panelists. Who's putting their hand up in the panel? Olga, go ahead.
Hi, sorry. Yeah, I put it in. I have to put it in the chat for some reason. It's just a very basic question. How do the endothelial cells or within the vascular system or even the vascular system itself affect the growth of the dendrite, neuron dendrites?
Yeah, sorry, I interrupted you.
No, I was just saying, because as you probably already know, SYNGAP1
yeah.
indicates early maturation of the dendrites. They grow too quickly and create chaos.
Yeah, so if we go earlier during development, in those niches, what we call the actually vascular niche of neurogenesis, you have those places in the brain where those neuronal progenitors develop and they are going to migrate at their final position. Without a healthy endothelium, this is not happening. So I'm going to hypothesize. Of course, we call it cell autonomously, meaning that inside a cell that is deficient for SYNGAP1, half the expression, for instance, this is going to trigger intrinsically some deficits that are reducing formation of those elongations and dendrites. However, what I think is in part, if you lack a proper endothelium, particularly during developmental periods, this could delay or misinstruct this neuronal progenitor to go to a specific fate and definitely affect their differentiation, their migration, for instance, or their division. So I don't think that vascular features would affect the way that SYNGAP1 itself is already influencing neuronal morphology, but I would see it as a regulator. If you lack what we call the angiocrine function, the release of molecules that are regulated by the endothelium, if you mess up the system, I think there might be a dynamic between those two, and that's one of the things we'll look at, because we can look at embryonic brain in mice. We could look at phases of development throughout life and ask these questions.
Okay, thank you. And one other quick question that I didn't get on here was just from a parent perspective, are there any specific natural supplements that you would recommend, or is that?
No, so even if I had an idea in mind, I would not recommend anything.
Okay.
First of all, I'm not an MD, and I don't want to.
Sure, cross that. I understand that.
Okay. Something wrong, but I hope we can find some.
Yes, thank you. It's very helpful.
Yeah, this has been fabulous. Thank you so much, Professor Lacoste, and we'll be in touch.
Yeah, thank you so much, everyone. It was a great pleasure to discuss with you all.
We appreciate it. Okay, goodbye, everybody.
Bye.